Okay, good morning. My name's James Stager. This is the second in a series of videos I'm making on articles published talking only about euploid embryos. So by way of introduction, if you're an IVF staff member, euploid embryos are only determined after PGT-A. They do represent viable embryos for transfer or storage. They may be either euploid or minimally mosaic embryos. They are identified by the biopsy of the trophectoderm and not the inner cell mass. They may not truly represent the future health of the baby, but they do show a 50% lower incidence of miscarriage after transfer and a shorter time to pregnancy. If you're an IVF client attending an IVF centre, the key things to know about euploid embryos is that about 40% of all embryos that clinics make have the correct number of chromosomes. These are called euploid. They can only be determined after biopsying of the embryo, called PGT-A. It's important that euploid embryos, which are normal, have a lower chance of miscarriage. But however, some embryos labelled as not euploid, called aneuploidy, may also overestimate the likelihood of some inner cell mass being normal and the baby being abnormal. Some embryos are a mixture of euploid and aneuploid cells, called mosaic, and the degree of mixture can reflect the health of the future child. As a general rule, embryos with less than 40% aneuploid cells, may be transferred, but if more than 40% cells are aneuploidy, they are normally discarded. Okay, the 5 papers I wanted to talk about today. One looking at the inner cell mass grade and early blastulation and the likelihood of pregnancy. The second one's looking at the size of the expanded blastocoel cavity. The third is looking at the developmental rest rate of embryos in relationship to aneuploidy. Looking at the fourth one is euploid embryos that fail to implant. Looking at trophectoderm transcriptomics of the left and seeing whether that can make a difference. And the last is looking at the meta-analysis of sperm DNA fragmentation on embryo euploidy rates. Okay, so let's start. First one, published in Reproductive Science in October this year, looked at the inner cell mass grade and the time of blastulation in relationship of euploid embryos in relationship to pregnancy. So only looking at euploid embryos. The first thing they reported was that even where the gender of the embryo was known in about 90% of cases, in reality, half the embryos transferred were male and half were female. So basically, just looking at the quality of blastocysts, you can't tell the difference between male and female. But even in cycles where the sex was used to select embryos for transfer, that occurred in 52% of cases and 70% of cases where they were female. And basically, you can say that the embryo gender, the sex, whether that was used for sex selection or not, were not associated with the odds of a live birth. However, when you go back and look at the embryo quality, you can look at the day of vitrification, that is how fast the embryo was developing. Quality of the inner cell mass was affected in both male and female embryos. You can see here that embryos that were growing slow, male embryos that were growing slow, had a significantly lower chance of pregnancy compared to day 5. But the quality of the inner cell mass which affected them in this group were not significantly associated with a live birth. And interestingly enough, when you look at female embryos, the day of vitrification, day 6 and day 7, were not associated with any increase or decreased chance of pregnancy. But the quality of the inner cell mass on quality from good to poor was highly associated with the chance of live birth. But if you look at the quality of the trophectoderm, trophectoblast, as they say, 2 to 3 was not associated with pregnancy. So basically summarising all that, if embryos that were developing fast on day 5 had a good quality inner cell mass, the ICM grade 1 is equivalent to Gardner grade A, and you had a female embryo, the chance of a live birth was 61%. That's about as good as it gets. Male embryos were about 4% less, but that's not really significant. However, if the inner cell mass quality is poor, their ICM2s are equivalent to Gardner grade B and C. The chance of live birth drops from 60% down to about 42%. And not much difference between male and female. However, if the embryo takes a bit longer to form, even with on day 6 with a good quality inner cell mass, the pregnancy rate 55% is almost no different to the day 5. The male embryos, however, in this group were not quite as successful. So there were some differences in male and female embryos, even with good quality inner cell mass, if the embryos still take a little bit longer. And I think this reflects the observation that male embryos really like to grow fast. And when they're growing slow, that's a problem. That tells you something's wrong. And if you have embryos that took 6 days to get to blastocyst and have had a poor quality ECM, both male and females had a live birth rate. Still 36% is pretty good, but only half what you would have expected if they got there on day 5. So in conclusion, euploid embryos with a better ICM quality and those that reached the blastocyst stage earlier were associated with higher odds of live birth, suggesting that prioritizing embryos for transfer by these parameters may provide patients with the best chance of a live birth. Male and female embryos may not behave identical, but in terms of live birth, they appear to have similar outcomes. Male embryos may develop faster, but the ICM may take a little bit longer to develop. Okay, so just to summarize what the author said, they found that both the day of vitrification and the quality of inner cell mass at vitrification were significantly associated with the odds of live birth in euploid embryos. In contrast, the quality of trophectoderm, or what they call a trophoblast, at vitrification, the sex of the embryo, and whether the embryo was selected on basis of the gender were not associated with live birth. And repeating ourselves, I think, I apologize for that. In conclusion, euploid embryos with a better ICM quality and those that reached the blastocyst stage earlier were associated with higher odds of live birth, suggesting that prioritizing embryos for transfer by these parameters may provide patients with the best chance of live birth. Okay. The next paper from Taiwan looked at the role of the size of the blastocoel cavity in relationship to the ploidy status of the embryo. And they concluded that the blastocyst inner diameter showed no significant association with ploidy status post-hatching after PGT-A. Euploid embryos reach full expansions faster than aneuploidy and mosaic embryos, and low mosaic embryos resembled euploid, unlike high mosaic ones. So you can't really tell the difference in rates of development between euploid and low mosaics. They're almost identical. Therefore, the time to hatching and the day 5, day 6 kids score could aid in selecting high quality embryos for genetic testing post-assisted hatching. In this study, they examined the time interval from fertilization to hatching, the inner diameter, and the kids score day 3 and day 5 for euploid and euploidy mosaic embryos, and they found that the aneuploid group had significantly longer time to hatching and a lower day 5, day 6 kids score compared to the other two groups. And just looking at the data, so the trophectoderm grade A, B, C, these are Gardner's grade A, B, and C. And so you look at the distribution, the majority of the embryos here were what they call a B grade trophectoderm. The time taken, so if an A grade trophectoderm, it took about 105 hours to get to that, whereas the B and C grade took 11 or 118 hours to get there. And that was significantly different between the blastocyst, the trophectoderm quality. The diameter made no difference at all, but the euploidy rate was highly significant, where the euploidy rate for A grade trophectoderm embryos is about 45%, dropping to about 30% for B grade, and falling even further precipitously to 16% for C grade embryos. That's probably self-evident. But interesting, for the inner cell mass, the embryos that an A grade inner cell mass, that is where there's a clearly defined inner cell mass with lots of cells and fairly firmly attached to the side of the blastocyst. Majority of embryos only had a B grade inner cell mass, but the ones that had a really well developed inner cell mass, they got there in a shorter time than B and C grade. The diameter once again had no relationship to the inner cell mass, but the euploidy rate, like the trophectoderm, the embryos that had an A grade inner cell mass had the highest rate of euploidy, 47%, dropping significantly down to 28%, almost half with B grade, and half again with very little inner cell mass, highly significant. So just summarising it for your euploidy and aneuploidy mosaic embryos, the majority of embryos in this group were aneuploidy, but you add the euploidy mosaic together and you get kind of 50 odd. The KID score between the three types was not significant, but the day 5 KID score was. So the euploidy embryos had a KID score of 6 out of 10, and the aneuploidy 5.7. There's not a lot of difference in that, in all fairness, but it was significant, and the mosaic embryos were somewhere in between. And likewise, the time to hatching, just under 110 hours for euploid, 114 for aneuploidy, and 110. So there's a significant difference between them, but the euploidy and mosaic embryos were behaving much the same. And in relationship to the paper, the diameter of the blastocyst was the same regardless of whether they're euploidy or aneuploidy. The date with the embryo was biopsied on day 5 or day 6. The majority of euploid embryos, 80%, were biopsied on day 5. And for aneuploid embryos, half were biopsied on day 5 and half day 6. So if you're growing an embryo out to day 6, you suddenly go from a 4 in 5 chance of being euploid down to about 1 chance in 5. So a comment here is that euploid is related to day 5, day 6 KID score, time to blastulation, and suitability for biopsy on day 5. The euploidy was not related to day 3 KID score, the diameter, and I can make a comment, even though the day 6 biopsies can result in euploid embryos, they're more likely to result in aneuploid mosaic. It's a lot of extra work for a diminishing gain. The next paper is one looking at why euploid embryos may not implant. So this group from the Spanish group looked at the transcriptomic differences in the mural trophectoderm between euploid blastocysts on the basis of their ability to remain attached in extended vitro culture and to identify gene expression profiles associated with competency for implantation. It's a prospective in vitro experimental cohort study involving ribonucleic acid sequencing of mural trophectodermal biopsies from euploid blastocysts and extended culture up to day 11. There's a reference. So basically, this had 15 euploid embryos donated by 16 couples undergoing ICSI and pre-implantation genetic study for aneuploid testing. The transcriptomic comparison focused on 10 euploid blastocysts classified as attached 5 or unattached 5 after extended in vitro culture, and the rest were excluded for various reasons. So we're only talking about 10 embryos. But what they could show, and since this paper was not open access, you can't see all the data, unfortunately. So much stuff is lost by not being open access. The study demonstrated that mural trophectodermal gene expression differed significantly between euploid blastocysts that attached and those that did not attach, highlighting molecular pathways potentially linked to implantation success. And the authors make a note that despite human implantation typically occurring through the polar trophectoderm, biopsying the mural trophectoderm transchromatic profiles may provide predictive value for embryo selection and deepen our understanding of early implantation biology. They need to get further validation. And I just want to make a comment that this is part of a group of studies now looking at RNA activity in embryos or spent culture media, and one can see it eventually leading to further trophectodermal analysis after PGT-A apart from chromosome analysis. And there's no doubt in the future it will explore this or polygenic scoring to value add the PGT-A process. This is probably 1 or 2 years away. The next paper is just looking at sperm DNA fragmentation on the euploidy rate. This was from Chinese group, I think, open access. It's actually quite a good paper. And they basically want to quantitatively evaluate the association between sperm DNA fragmentation and embryo euploidy rates in IVF cycles through a systematic review and meta-analysis. I have to say they looked at 1,500 papers, and of those they selected 6 that were suitable. So that's almost 1,500 papers on IVF, and euploidy were actually discounted because of the structure of the paper. It does suggest that when you're looking at these things, you need to be looking to write a paper where it can be part of a meta-analysis. They're all looking at SCARSA tests, and they looked at, there's a variety of them. In fact, you can go back, I've made a separate YouTube video on this particular paper, which will be uploaded shortly. But if you compare really good quality sperm, less than 50% DNA, to more than 50%, it's not significantly different. You see the ratio is overlapping the even bar. If you then compare high DNA fragmentation to moderate samples, you can see there's a trend here, but it's still not significant. But when you compare samples with more than 30% DNA fragmentation to less than 30% DNA, there was a significant association. Some tests show a very strong group. This one here from Gayos, it's almost in the odds ratio positive range, but there are two papers there which actually still don't find the association. So they're still very much work in progress. So they concluded the elevated sperm DNA fragmentation, particularly above 30%, is associated with reduced likelihood of obtaining euploid embryos in ART cycles, suggesting a potential threshold-dependent effect on sperm DNA integrity on mammalian, on embryo chromosomal normality. These findings support the integration of SDS assessment in the evaluation of selected couples, especially in cases of recurrent ART failure or advanced maternal age. And that, my friends, is the key statement. I don't need to do that one. The next paper, I think, is an interesting one. That's looking at the arrest of embryos, the developmental arrest of embryos in relationship to age and euploidy. Does the aneuploidy in an embryo cause some embryos to stop developing? So this study in aging, why would you ever find an article published in aging? But it's a good paper, though. This study aimed to investigate the extent to which developmental arrest rate in embryos generated by IVF correlate with female age and the rate of aneuploidy in the cohort. And overall, so you can see here the main graph, these are the different age groups. The start groups from America, 30% under 35, 35 to 37, 38 to 40, 41 to 42, and over 42. This is the rate of developmental arrest, and these are the subgroups of how much aneuploidy. So this one, for instance, here is more than 75% of the cells are aneuploidy compared to less than 50. Here, for instance, it's less than 25% of the cells are aneuploidy compared to 75% aneuploidy, and looking at the rate of arrest. So they reported a very weak positive correlation was identified between developmental arrest and the rate of aneuploidy right across the whole group. But when you adjusted for age, no significant difference did exist. So most of the effect was due to age. All the included cohorts were divided into the Society of Assisted Reproductive Technology START age groups according to the patient age, as I've indicated before. The findings suggest that the rate of development arrest and the rate of whole chromosome aneuploidy in the resulting blastocyst cohort are both associated with female age and the amount of aneuploidy, but not with each other. Therefore, early embryonic development arrest and aneuploidy rates represent two independent factors in determining the number of euploid embryos that are available to transfer and the overall likelihood of success. So in summary, you can say that euploid embryos with better ICM quality and those that reached the blastocyst stage earlier were associated with higher odds of live birth, suggesting that prioritising embryos for transfer by these parameters may provide patients with the best chance of live birth. Euploidy was not related to day 3 KIDS score or the diameter of the embryo. The study looking at mural trophectoderm gene expression differed significantly between whether the euploid blastocysts were able to attach and those that could not, highlighting that there's a molecular pathway potentially linked to implantation success. Another study also showed that elevated sperm DNA fragmentation, particularly above 30%, is associated with a reduced likelihood of obtaining euploid embryos in ART, suggesting a potential threshold-dependent effect of sperm DNA integrity on embryo chromosomal normality. I think in this respect, and I said I've discussed this more in a separate YouTube video, is that most of the aneuploidy in embryos is due to the oocyte. The sperm contributes only about 5 to 10% aneuploidy. And I suspect most of it is to do with aspects of the damaged sperm and increased levels of mitotic errors as the embryo divides. So most, I think, is largely to do with chromosomal, with mosaic embryos. And finally, the rate of embryo development arrests and the rate at which whole chromosome and aneuploidy in the resulting cohort are associated with female age, but not with each other. So that, my lot, is your lot for the week. My name is James Stanger, an embryo with more than 50 years experience. And a disclaimer, I'm not a medical person. This information is not medical advice, but it may help you in knowing what questions to ask about your treatment with your doctor, and maybe to help you understand the advice you receive. If you like what I'm doing, click subscribe, leave a comment, and always press like. And just a quick plug from my other business activity, which is ferday.com, which is an online EQA and training program in andrology and embryology. Looks at a variety of different schemes in andrology and embryology, all done online. You can enroll as a trainee or as a professional. You can do small clinic or large clinic laboratories, and you can use it for training or competency or for accreditation. Okay, enough of that. So I'd like to thank you for your patience if you got this far. I understand IVF is a personal journey that involves the action of many different techniques and staff, and it helps to understand all the aspects of that journey. I hope this information may be of value. If you like it, please tell other clients about the service and please subscribe. At that point, I think I'll say au revoir.